So I'm going to be talking about the Hershey-Chase experiment. So in the Hershey-Chase experiment, there are two batches. I'm going to start with batch 1. So in batch 1, Hershey and Chase grew T2 with E. coli in a solution containing radioactive sulfur. That's right here. And so the phages contain sulfur, but the DNA does not. And so in step 1, they mixed radioactively labeled phages with bacteria, and the phages are going to infect the bacterial cells. And in step 2, they agitated it in a blender to separate phages outside the bacteria from the cells and their contents. So we can see here that the bacteria, the DNA, has gone inside the bacterial cell, and then here we have empty protein shells. So the third step is to centrifuge the mixture so the bacteria form a pellet at the bottom of the test tube. So here we have our test tube, and we have the liquid here, and then we have the pellet here. And we can see that the radioactivity ended up mainly in the liquid, and the liquid contains phages but not bacteria. So this shows that the phage protein never actually entered the bacterial cell. So batch 2 is conducted in the same way, except Hershey and Chase grew T2 with E. coli in a solution containing radioactive phosphorus. So the DNA of the phages are green and contain phosphorus, but the protein does not. So after agitating in a blender and centrifuging the mixture, we have our test tube here, and we have our pellet and our liquid. So we can see that most of the radioactivity is found in the pellet, which shows that the DNA was actually able to get into the bacterial cell. So Hershey and Chase were able to conclude that T2 injects its DNA into the host cell, leaving virtually all its protein outside. And we know that because it's all the protein ended up in the liquid and not in the pellet. So yeah.